Hi everyone, welcome to Spatial Omics Seminar Series 5. And today we are very happy to have Dr. Rong Fan from Johns Hopkins University. And briefly, Dr. Rong Fan received her PhD in the Bioinformatics and Integrative Genomics program from Harvard Medical School in 2018. Then she performed her postdoctoral fellowship at the Chemistry and Chemical Biology and Physics Department at Harvard University with Xiaowei Zhuang. And after 2020, she has started her lab at Johns Hopkins University, and she's currently a biomedical engineering assistant professor. And also she's part of the Center for Computational Biology at Johns Hopkins. And he recently also received, we were just chatting about this earlier, NSF career for her work on statistical spatial transcriptional analysis. And with this, I think I'd like to turn over to Dr. Fan for learning more details.
Perfect. Well, thank you so much to everyone for being here, and to Ahmet and Rong for the invitation and opportunity to teach you all a little bit about our work on developing statistical approaches and computational tools for analyzing spatially resolved transcriptomics data. And let's see if I can get this to progress. There we go. Yeah, so just before I get started, I just wanted to give a quick shout out to my lab and just highlight some of the friendly faces behind the work you're about to see. We do call ourselves the JeffWorks Lab to distinguish ourselves from the other prominent fan lab. As Ahmet mentioned, we started last July and we're coming up on our 1-year anniversary, but we're still actively looking for collaborators. So please do reach out if you're interested in getting a conversation started there. Okay, so our lab is generally interested in understanding the role of subcellular and cellular spatial organization and how that shapes cellular identity and heterogeneity. So in other words, particularly for polarized cell types like the neuron here, perhaps cells undergoing differentiation and division, we're interested in understanding how mRNAs are organized within the cell. And likewise, in the context of, say, tumors, where we have many transcriptionally distinct neoplastic subpopulations that might be interacting with non-neoplastic cells in the tumor microenvironment, we're interested in understanding how cell types and cell states are organized within the tissue. And then ultimately, how does this spatial organization end up relating to downstream functional consequences? So luckily for us, there have been quite a number of advances in sequencing and imaging technologies that have really enabled us to profile in a very high throughput manner these spatially resolved transcriptomic measurements at either subcellular, cellular, or even pixel resolution. So these are just a few of the technologies highlighted. We know DiviTseq from Rong's group, and I think a few weeks ago, Jeff gave a presentation on MERFISH. And there are many, many more technologies that I think every time I present this, I feel the slide becomes more and more outdated. But generally, these technologies will differ in their chemistry, their resolution, their throughput or sensitivity, or error correction schemes, and so forth. But ultimately, what they allow us to do is allow us to measure precisely what genes are expressed at what spatial locations, and to do this for let's say hundreds to full transcriptome numbers of genes, and in of course hundreds to thousands, if not millions of molecules inside cells, inside pixels. So then, of course, given this amazing spatially resolved transcriptomics technologies, we really need new statistical approaches and computational tools for analyzing the resulting spatially resolved transcriptomics data. So for those of you interested in learning a bit more about the particular computational challenges that we envision moving forward, my student Lila Atta and I are contributing an invited commentary on this topic. So please do be on the lookout for that in Nature Communications if this is of interest to you. Okay, so as I mentioned, there are many technologies for spatially resolved transcriptomic profiling, one of which is called Multiplexed Error-Robust Fluorescence in situ hybridization, or MERFISH for short. And what MERFISH allows us to do is achieve this type of spatially resolved but targeted transcriptome imaging at single molecule resolution with a very high detection efficiency and a very low error rate. And since Jeff spoke at length about this a few weeks ago, I'll try to skip the details of how MERFISH works. But if you're interested, please do check out the original Chen et al. 2015 science paper. And what effectively, what MERFISH allows us to do is take a picture of where different RNAs are inside us, inside fixed cells. So here we have, a cell, and each point is a different molecule, and we can precisely image, or decode after many rounds of imaging, what molecules correspond to gene A, what molecules correspond to gene B, and likewise for hundreds to now up to 10,000 different targeted gene species. And of course, we can do this not just for one cell, but we can do this for a large field of view with many cells, or even tile across many fields of views to be able to make these spatially resolved transcriptomic measurements for many cells. And then we can actually segment, computationally segment these cells and count, how many of each gene is within each cell, and in that manner achieve a single cell resolution spatially resolved transcriptomic profiling. Oops. Go forward. Okay. So having this type of rich spatially resolved transcriptomics data, we can go back to our original biological questions of interest and see if we can derive new insights regarding, for example, how our mRNAs are organized within the cell. So this was work in collaboration with Chenlong Xia and George Emanuel and Xiaowei's lab back when I was a postdoc. And this is work that has been published in PNAS in 2019. But here what we wanted to do is ask, what mRNAs are organized in different subcellular compartments, such as the nucleus versus the cytoplasm. And in particular, Chenlong used MERFISH with expansion microscopy to profile over 10,000 genes in about 1,000 cultured U2OS cells. But because, this is an imaging-based approach, we can actually then computationally segment the region of the cell that corresponds to the nucleus and count how many of each different gene species is within the nucleus compared to the what genes are within the cytoplasm, and do this for all our cells and end up with something like this, where we have a quantification of each gene's expression within the nuclear compartment and also within the cytoplasmic compartment for all the cells that we have. And for those of you who are perhaps more familiar with other types of differential expression or bulk transcriptomics analysis, for here, rather than asking what genes are upregulated and say cancer versus healthy and paired tumor normal samples, we can instead ask what genes are enriched in the nucleus versus the cytoplasm. And we can essentially take advantage of all the lessons we've learned from doing this type of differential expression analysis in the bulk RNA-seq world, like controlling for potential covariates and library size and so forth. But what we get at the end is this, it's very similar to what you might expect from another type of differential expression analysis, where we get these volcano plots where each point is a gene, and we can see that there are some genes that have very high fold change and very significantly enriched within the nucleus. And given that these are imaging-based approaches, it's very nice to then be able to visualize our results on the original cells and see that indeed the genes that we identify as being significantly enriched in the nucleus do spatially co-localize to the nucleus. And of course, for these genes, some of these genes, we know them to be enriched in the nucleus, like long non-coding RNAs or intron-retained genes. But interestingly, we also do find a large number of protein coding genes that seem enriched spatially within the nucleus. And so now there's ongoing work in the lab to see just how this spatial, subcellular spatial organization might ultimately impact the eventual protein expression levels of these genes, and see how nuclear retention of these protein coding genes may be used as a mechanism to modulate protein expression in these cells. Okay, and of course, we can also look at more polarized cell types such as neurons, and ask, how are RNAs or mRNAs organized within, say, the soma versus the dendrite versus axon, or even a long axons. So this was work really spearheaded by Guiping Wang, and she applied MERFISH with expansion microscopy to characterize over 4,000 genes in cultured hippocampal neurons. So again, we can identify genes that are enriched within the dendrite compared to the soma, but we can also take inspiration from single cell analysis and actually do clustering analysis, where now each dot here, rather than being an individual cell, is actually a segmented soma or a dendrite. And in this manner, show that through clustering analysis that the soma and the dendrite are actually quite transcriptionally distinct, that it's not just a few genes that are, let's say, enriched here and there, but they really are very transcriptionally distinct entities. And these two clusters actually correspond to inhibitory and excitatory neurons. But so there are transcriptional differences. But then we can also see, for example, not just between soma and dendrite, but even along the dendrite itself, as a function to the distance to the cell soma, are there certain genes that are enriched, at the proximal regions, at the more, middle-ish regions, or versus the more distal regions of the dendrites, and identify just interesting spatial patterns that are actually shared across both inhibitory and excitatory neurons. So I think overall, we do hope that this type of high-resolution spatial atlas at the subcellular level within neurons could be a valuable resource to the community. So please do check out Guiping's paper on bioRxiv. Okay, great. Well then, jumping a little bit, out of the subcellular spatial organization world, we're of course also very interested in how cells and cell states are organized within tissues, particularly, as I mentioned before, in the context of the tumor microenvironment. So this is work done in collaboration with Toshi Hara and Mario Suva's group, as well as Itai Roche's group, with Lila Atta and my group leading the spatial computational analysis. But we really wanted to address this question of how are transcriptionally distinct neoplastic subpopulations spatially organized in tumors, in this case in glioblastoma. So previously, Mario's lab used single cell RNA sequencing to characterize IDH wild type glioblastomas to identify these 4 transcriptionally distinct subpopulations in glioblastoma. So, we have more OPC-like glioblastoma cells, more astrocytic-like, more neural-like, and more mesenchymal-like glioblastoma cells. So we can see that from the single cell sequencing data. But what we can't see is how these subpopulations are spatially organized, particularly with respect to other cell types potentially within the tumor microenvironment. So therefore, we took on an application of MERFISH for just a select panel of 100 or so genes to try to distinguish between these 4 different glioblastoma subtypes, as well as major cell types within the tumor microenvironment. And what we can do is, with this data, we can do clustering analysis, just as we typically do in single-cell RNA sequencing data. But here, of course, we only have 100 genes. So we can just show that, even given this 100 genes, we can distinguish, our oligodendrocytes from our macrophages, from T cells, and distinguish between the mesenchymal subtype of the glioblastoma cells and so forth in a manner that's very consistent with single-cell RNA sequencing. And likewise, just confirm that in this case, each point is a single cell, either from MERFISH or from single-cell RNA-seq. And you can see that the two technologies, this is analysis with harmony, but generally we can identify the same clusters of cells. And of course, when we look at the transcriptomes of these cells for the 100 or so limited genes that we have, transcriptionally, the macrophages we identify from the MERFISH data is quite transcriptionally similar to the single-cell RNA-seq data. So then we can use this as a way of transferring our labels from our single-cell RNA sequencing onto our MERFISH. But then, the fun thing with spatially resolved transcriptomics technologies is that, we can identify these transcriptionally distinct subpopulations of, different cell types and cell states. But then we can see how they map back onto the original spatial coordinates. So now we can actually ask, our biological question of interest and test, whether different neoplastic subpopulations, for example, spatially co-localized with different immune cells or other cells in the tumor microenvironment. And interestingly here, what we observe is that when we look among the spatial neighbors of the mesenchymal-like glioblastoma cells, we see a lot of macrophages and more macrophages than what we expect by chance based on just a permutation null model. In contrast, we don't see the same type of spatial enrichment for oligodendrocytes, for example, in the spatial neighbors of the mesenchymal cells. And likewise, we don't see this type of spatial enrichment for macrophages among the spatial neighbors of the other glioblastoma subtypes. So this seems to be a type of spatial enrichment unique to this mesenchymal subtype and unique to macrophages. So then in parallel, Toshi Hara and Itai Teroshi's team were able to do a lot of interesting work with xenograph models and other analyses with single cell sequencing data to discover that these macrophages are actually inducing a particular transition of the glioblastoma cells into this mesenchymal-like state via this particular macrophage just derived oncostatin M that interacts with the corresponding receptor on glioblastoma cells. So I think this is just overall a really beautiful example of how, in this case, we have this spatial organization of macrophages and mesenchymal glioblastoma cells that end up actually having a functional consequence. So for more details on this paper, please do check out our cancer cell paper, which I think came out just a few weeks ago. Okay, great. So we're very good on time. So then the last analysis I really wanted to teach you all about a little today is regarding this question of how can we identify spatially heterogeneous gene expression within tissues, particularly when there's a lot of variability in cell density within tissues, and likewise, can we identify spatially heterogeneous gene expression patterns that are potentially orthogonal to the underlying spatial organization of transcriptionally distinct cell types. So for this type of analysis, we drew on inspiration from geospatial analysis, where there's this statistic called Moran's I, which has been used to evaluate whether a particular quantity, say, in this case, income, let's say, exhibits significant non-random spatial variation, or also called spatial autocorrelation. In this case, even though this looks kind of like a tumor, I suppose it's actually I guess a state, and these little blobs are counties, and the colors are denoting income level. And so, what you can just visually appreciate is that income is not random. It's actually the income of, say, this county here is highly correlated with the income of its neighbors, of the neighboring counties. So then in this particular case, people have in geospatial analysis have used Moran's eye to quantify that income exhibits very high spatial autocorrelation, that it's not random. And of course, we can talk about the perhaps political and historical reasons for this, but at least today we are interested in not income but gene expression. So we can think of a similar situation where now rather than having counties, each point is a single cell, and sums for this particular gene, red is high, blue is low. You can see that this gene's expression in this cell is always correlated with the gene expression in the neighboring cells. So this Moran's eye statistic will be positive. And in contrast, a gene, for example, here, where it's high expression in this cell, but it doesn't seem that correlated with the expression levels in the neighboring cells. So the eye statistic will be low. And in this manner, we can identify genes that distinguish genes that either exhibit significant spatial variation or have a variation that is not coupled with the spatial organization. So we developed this R package called Meringue to play on the name of Moran and MERFISH, and it also happens to be one of my favorite desserts. But we build on this spatial autocorrelation analysis to specifically identify genes with spatially heterogeneous expression patterns, and also infer putative cell-cell communication, but focusing on situations where we're dealing with non-uniform cellular densities, which we expect within tissues. And this is work with my student Brendan Miller in my lab, and a lot of insightful input from DJ Mabamuku and Catherine Dulac particularly on the biology of things. So for more details on this method, please do check out our paper, which is coming out in Genome Research. But I believe there's an accepted unformatted version of the manuscript already available. Great. So just briefly to explain what Meringue does, it's identifying spatially variable genes by calculating the spatial autocorrelation statistic. But there's actually many ways of calculating it that relates to how we encode adjacency relationships via this W matrix, which we call an adjacency weight matrix. So in general, we can interpret this W matrix as encoding spatial relationships where each entry in the matrix if you have n cells, it'll be n by n matrix. And if two cells are neighbors with each other, then this W for cell I and cell J, if cell I and cell J are neighbors, then this W value will be 1 and 0 otherwise. So if we stare at this equation a little, what you might see is that, if, let's say, the gene expression level for, encoded by the variable X is high in, cell I, and it's also high in cell J, and I and J are neighbors, then, this number will be positive and large. So then the I statistic will be large. And likewise, if they're always low together, then, I statistic will also still be positive. But if if I and J are not neighbors, then W will be 0, and it actually contributes nothing. But yeah, in general, if the value of X is highly correlated between that of its spatial neighbors, then this I statistic will be very positive, and we can evaluate the statistical significance of that by solving the expected value and variance for this particular equation. But actually, how do you define who are your spatial neighbors? Well, it turns out that if we have this situation of a very non-uniform cell density, as we commonly see in the tissues, for example, this is simulated data, but each point is a single cell, And some areas have high density and some areas have low density. That's just how cells are naturally within tissues. But in this particular case with the non-uniform cellular density, if we define spatial neighbors based on just distance, what we find is that even if we have completely random gene expression, such as either generated through Gaussian noise or generated by randomly permuting real spatial gene expression data, what we find is that even in this, even when given random genes, because of this confounding of the cell density, what we end up with is a very high likelihood of picking up these random genes as being significantly spatially autocorrelated. So we can look at the distribution of the p-values and try to get at under this random gene expression model and try to get at a false discovery or a type 1 error rate, which for distance-based neighbors can be quite high. And likewise, if we choose neighbors based on k nearest neighbors, depending on what k we choose and depending on the extent to which our cells vary in their densities, we can again get this very inflated type 1 error rate. However, by defining cell neighbors based on Voronoi tessellation, we end up with this much better uniform null distribution, which gives us the expected type 1 error rate. So therefore, meringue uses this Voronoi tessellation-based approach for identifying spatial neighbors. It's sometimes also called Delaney triangulation, where again, each point is a single cell, but now we can represent these cells as neighbors if their Voronoi tessellations share an edge. And this particular way of defining spatial neighbors is quite flexible to distortions in cellular densities, but it's also amenable to both 2D structures and also 3D structures. And one additional thing that we've noticed when we try to borrow from these geospatial analyses to the biological domain is that not all spatially heterogeneous gene expression patterns are equal. In biology, we are often interested in not just spatial patterns that are driven by a single hotspot, for example. We often would like to see spatial patterns driven by a larger percentage of cells, for example. So then what meringue does is build on another geospatial statistic called local indicators of spatial autocorrelation, or LISA, to characterize the scale of the spatial patterns. So it's no longer, we don't look for just significant spatial autocorrelation, but also spatial autocorrelation that is driven by a sufficient number of cells or sufficient percentage of cells. And using this additional criteria, what we can show is that again, under random, evaluation of random noise, if we compute the percentage of cells, driving a particular spatial pattern, if we require the spatial pattern to be driven by, more than 5% of cells, that effectively eliminates, all our false positives. So these are the frequencies of genes that are random genes that are detected as significantly spatially autocorrelated versus the percentage of cells driving that particular spatial pattern. So if we just use a threshold of 5%, we can get rid of all of this. These are all considered false positives, essentially. Great. So then, as a proof of concept, we wanted to apply meringue to identify spatially heterogeneous genes in the mouse olfactory bulb, or MOB. I guess I'll call it MOB. So the MOB consists of multiple transcriptionally distinct cell layers, as you can see here. So then, given spatially resolved transcriptomic profiling, we can, of course, cluster and identify these transcriptionally distinct cell clusters. But when we map these clusters back onto their original spatial coordinates, we can see this very beautiful, bilaterally symmetric layer structure that is consistent with, the spatial layers that we know to be present in the MOB. Expectedly, in this particular case, given that these transcriptionally distinct cell layers are organized spatially, we would expect, if our approach works, that we would identify spatially heterogeneous genes that are consistent with these cell layers. And indeed, that's what we find. When we apply meringue, we can identify significantly spatially autocorrelated genes driven by more than 5% of pixels, and all the genes we identify significantly overlap with differentially expressed genes across the different cell layers. And likewise, of course, we can compare our method to other existing methods for identifying spatially variable genes and find that the negative log 10 adjusted p-values for our approach is very, very highly correlated with that of other approaches as well. So because meringue encodes spatial relationships using this density agnostic manner via the Voronoi tessellations, even if we were to, for example, stretch or warp or distort these, in this case, we start out with a uniform cellular density, but even if we were to distort the cellular density into something that's no longer uniform, what we find is that we can still identify generally the same set of significantly spatially heterogeneous genes. So if we look at, each point is a gene, and the negative log 10 adjusted p-values under the uniform density are highly correlated with the negative log 10 adjusted p-values under the non-uniform density. Whereas in contrast, other approaches, especially understandably, those who rely on using Euclidean distances between cells, they are much more sensitive to these changes in cell density. And if we distort the data and induce a non-uniformity in cell density, they are no longer able to identify the same set of spatially variable genes. And as I mentioned previously, this all comes down to the way that we define this adjacency weight matrix W in the Morazai statistic, which can be very flexible. We can do Voronoi tessellations in 3D, such as using in this example with a 3D Drosophila embryo, where we actually have the 3D spatial coordinates for every cell. And we can just define the cell's spatial neighbors in 3D rather than 2D, and in the same way still be able to pick up on significantly spatially autocorrelated genes. But we can also accommodate multi-section data, which often is a little distinct from true 3D data, since the sections tend to be a little bit more further spaced apart, for example. But we can identify, or we can define spatial neighbors exclusively to those that, to the cells that, for exple, align across these different sections rather than within sections. And in this particular manner, we can this particular manner of defining spatial neighbors allows us to look for spatial patterns that are, for example, consistent across tissue sections. So it's genes that exhibit spatial autocorrelation, but in a manner that's consistent across tissue sections rather than within tissue sections. So for this example, we took 4 consecutive sections of a particular breast cancer biopsy and defined this spatial weight matrix based on cells that are adjacent across the tissues, and we're able to identify a number of significantly spatially variable genes that are consistent across the tissue sections. Okay, well, so then having shown that meringue can identify spatially variable genes in a density agnostic manner, what we really wanted to do was apply our approach to look for spatially heterogeneous genes in the hypothalamic medial preoptic area, which is an area where you can see there are certain regions that are higher in cell density, or certain regions that are lower in cell density, and so forth. But this is an area of the mouse brain previously characterized via MERFISH by Jeff and DJ. And they were able to do this for across many tissue sections from the anterior to the posterior MPOA. But this, and also across many animals, I should mention. But this MPOA region of the brain is particularly interesting because it has been previously shown to be essential for controlling social behaviors such as parenting, mating, and aggression. So we're very interested in just understanding how these various behaviors potentially may be molecularly encoded or spatially encoded. So Jeff and DJ previously performed clustering analysis on the resulting MERFISH cells, and were able to identify a large number of different cell types and also neuronal subtypes, and evaluate how they were spatially organized within this particular region. But what we wanted to do now is then ask essentially, even within each cell type or neuronal subtype, can we identify any additional aspects of spatial gene expression heterogeneity? So can we identify additional spatial gene expression variation within cell types, particularly those that were consistent across tissue sections and likewise reproducible across animals? So applying meringue to essentially systematically evaluate over 70 cell types in over 12 tissue sections for over a dozen animals, we were indeed able to identify some neuronal subtypes for which we could identify aspects of spatial gene expression heterogeneity. So for example, here we have each point is a cell, but the inhibitory I6 cells have been highlighted, and the expression of GAD1, for example, is being shown. In this case, GAD1 is actually a gene that we identified as being not spatially variable. And unsurprisingly, it's actually an inhibitory neuronal marker that's, as you can see, pretty uniformly expressed. So we wouldn't expect it to be spatially variable. But what we do find is there are other genes, such as NECAP1, which is a gene that encodes for a calcium binding protein, and likewise genes that encode for secreted glycoproteins that play a role in neuronal guidance, or even neurotransmitter receptors and so forth. These genes can, within the context of an individual cell type, can still exhibit prominent spatial or significant spatial heterogeneity, which visually you can already appreciate here, with red being high expression and blue being low expression. And sometimes these patterns are similar for different genes. Sometimes they're complementary. In the case of GAPA1 and OS1, they seem to mark different subpopulations of inhibitory I11 cells, for example. But all of these patterns are quite consistent across tissue sections. So if we look at the adjacent tissue sections, they still exhibit a similar type of spatial gradient. And likewise, they're reproducible across animals. But interestingly, well, not all spatial patterns were consistent across animals. There's this one particular gene, TACR1, which actually encodes for a neuropeptide receptor. Its expression in excitatory subtype E8 we did find as spatially significantly heterogeneous, but only in male animals, which you can visually see here. Or again, each point is a single cell, but the E8 cells are highlighted, and blue is low expression of TACR1 and red is high expression. And these are just a sample of 3 different female animals and 3 different male animals. And just visually, you can see that perhaps it turns out that there's no significant difference between males and females in terms of the proportion of cells, E8 cells that express TACR1, but there is a significant difference in their spatial organization, which you can just visually appreciate that the TACR1 E8 cells or the E8 cells that express TACR1 tend to be kind of aggregated spatially together in males, and it's more randomly dispersed in females. And likewise, we can appreciate this visually, but we can also quantify it using our Lisa statistic and just show that, even across different tissue sections, there is a consistently higher scale for the particular spatial pattern for TACR1 in E8 cells in a manner that's consistent across many animals. And so, again, given that the MPOA has been shown previously to control for essential social behaviors such as parenting, mating, and aggression, it's really interesting to see that perhaps these sexually dimorphic behaviors could be then related to not just the presence of different neurons at different proportions, but also the spatial organization of gene expression within neural subtypes themselves could be a potential way to functionally encode those sexually dimorphic behaviors. Okay, well then the final point I want to make is regarding how we can build on this Moran's eye statistic and modify it slightly, and again take advantage of the flexibility and the adjacency weight matrix definition to propose this new inter-cell type spatial cross-correlation statistic to look for genes that are cross-correlated, but among spatially co-localized cell types that could be indicative of cell-cell communication. So again, we have the original Moran's eye statistic where we are autocorrelating, which means we correlate X with itself for different cells I and J. So here we want to adapt that to now look for the expression of different genes, perhaps a receptor gene and a ligand gene, for example. And we want to limit this evaluation of this cross-correlation among not just spatial neighbors, but specifically spatial neighbors that are of different cell types. So again, we can modify this spatial adjacency weight matrix to only include cell types of cell type A and B, and only instances where the neighbor relationship is between a cell of cell type A and B and not of cell type A and A or B and B, for example. So then, considering this little toy example here, we might have, cell type A and cell type B, and there are some cells that are of cell type A and cell type B that are spatially adjacent to each other, and those would be the ones that we consider in our adjacency weight matrix. But then, for example, perhaps cell type A expresses a particular gene that encodes a particular, let's say, a receptor at different levels. And whereas, cell type B expresses this the complementary receptor, or the complementary ligand, sorry, at varying levels. So, given that the receptor and the ligand are expressed in different cell types, if we were to just look at regular cross-correlation between the gene expression of this receptor and ligand gene, we would actually find that it's negatively correlated. Because it's the receptor is only expressed in one cell type and the ligand is only expressed in the other. But of course, visually, we're smart. We can see that it just so happens as we simulated it, cells of cell type A that highly express the receptor are always spatially neighboring cells of cell type B that highly express the ligand. And so in this particular case, we can just quantify that interesting spatial pattern using our intercell type spatial cross-correlation statistic and show that indeed it is highly significant compared to what we expect under a random permutation null model. And in this particular case, this very significant positive intercell type spatial cross-correlation could be indicative of potential cell-cell communication between cell type A and cell type B via this particular receptor ligand interaction. So we just, having this particular statistic available, now we can apply it to our MPOA data. And we looked across various pairs of genes, and what we find is that interestingly, there is this significant and consistent spatial cross-correlation between a particular gene, this CYPP19A1 gene, which encodes for this enzyme called aromatase. But its expression in neuronal subtype I2 is significantly spatially cross-correlated with the expression of this ESR1 gene, which encodes for an estrogen receptor in the spatial neighbors of these I2 cells. And we can look across animals and look at the negative log 10 p-value. And generally, I2 is the only one that came out as significant across animals, though there is some variability across animals there. And in contrast, we don't see this type of similar significant spatial cross-correlation between aromatase and other genes, such as, say, the androgen receptor expression among the spatial neighbors for I2 cells, for example. So this appears to be a relationship unique to aromatase and the estrogen receptor gene. And just as a little bit of background, this is really interesting because aromatase is it's an enzyme that converts testosterone to estrogen, thereby modulating steroid hormone signaling in the preoptic region. And we all know testosterone typically binds androgen receptors, and estrogen binds estrogen receptors. So this particular spatial organization of aromatase expressing I2 neurons, spatially co-localizing with estrogen receptor neurons in the spatial neighborhood, could be suggesting that perhaps the estrogen being synthesized by these I2 aromatase expressing neurons may be interacting with the estrogen receptors on these spatially adjacent ESR1 expressing neurons in this paracrine manner. And I think this just all goes to show you that this is just another way that we can integrate spatial organization, in this case to potentially identify cell-cell communication, but ultimately relating this, these interesting spatial organizational patterns that we observe to potential functional consequences. Okay, well, yeah, so I just want to quickly summarize that. I hope I've been able to show you how spatially resolved transcriptomic data really demand new statistical approaches and computational tools to extract biological insights. And we can draw on inspiration from single cell analysis to characterize the transcriptional differences in different subcellular compartments. And likewise, we can draw inspiration from geospatial analysis to characterize gene expression heterogeneity. In particular, we developed a computational tool called Meringue to build on this spatial data analysis, specifically for spatial transcriptomics data in tissues to identify spatially heterogeneous gene expression patterns and also cell-cell communication in a manner that's robust to cellular densities or variation in cellular densities. And I think looking forward, there's just a lot that we can learn in terms of the biology as we can, as we begin applying these technologies, but also these computational tools to different biological systems, and particularly, in development and also in diseases like cancers. And of course, there's a lot that we can also learn from each other in terms of analytical techniques when we look across, these disciplinary divides. So with that, I just wanted to, if you're interested in learning how to use some of these tools or accessing different, spatially resolved transcriptomic data sets, we have a lot of tutorials available on our lab website for you to tinker around with. And yeah, with that, just thanks again to my lab members, and thank you all for your attention. Thanks again to our collaborators and our funding sources. And of course, you can always find us online, both on our lab website via email, and once in a while I can be found on Twitter. So yeah, thank you all, and happy to take any questions. Well, let's see, let me stop sharing. Let's see. Okay. think I can see you guys now.
It's a terrific talk. It's super exciting. I think a few questions there already. If you can.
Yeah, okay, let's see. Great. Edward Zhao asks, how are tissue sections aligned? Yes, that is a great question. For us, we are currently manually aligning. There are, I think, are more sophisticated geometric warping techniques that we are actually working to develop in collaboration with Mike Miller and Daniel Tord. But currently, we're doing very, very crude alignment across tissue sections that are just, very simple rotations and transformations. I think for a true alignment, there actually needs to be minor distortions within the tissue positions themselves because of variation in cross-sectioning and so forth. But yeah, that's a great question.
I think Danny gave a terrific talk last week.
Yeah.
That might be a potential solution.
Oh, exactly. Yes. Yes. Yeah, exactly. And yeah, as you saw, it wasn't sufficient to just align. You have to also do a little adjustments to get all the tubules to line. So yeah, that's exactly right. Let's see then. Let's see. Cole's Price asks, hey, awesome talk, awesome working presentation. Had a few questions. When using meringue, since it looks like Varina tessellation organizes cells as neighbors based on shared edges, if you take a cell-dense region like a tumor, how well does it perform in areas like that, where high regions of cell density, I assume, would automatically be a much bigger neighborhood? Are having neighborhoods with dramatically different sizes affect the analysis? And I know with K-based methods, like you showed, you need to define K ahead of time, but almost looks like you have to define your neighborhood in meringue, or maybe I understood that incorrectly. Yes, that's a great question. So the neighborhood actually, so we represent cells as points in space, and the neighborhood is defined by the Varina tessellation itself. So in regions where there are very dense cells, the neighborhoods would actually be smaller. So the way Varina tessellation works is you can think of it as almost like water filling a region, and it will expand out from the center of the cell until it reaches another cell, and then where that the two water flows reach is where the edge is. So in that sense, regions that are very dense, like in tumors, will have very, very small neighborhoods. And it's actually in this way, we actually are able to accommodate, neighborhoods that are smaller versus neighborhoods that are larger. But for very, very large neighborhoods, at some level, the cells are just too far apart from each other to interact anymore via, diffusion. There's kind of a length scale limit there. So we do have filters for, if your neighborhood is too large, it doesn't matter if you actually technically share an edge. If the distance itself is going to be too large for that spatial interaction. So there are some filters based on that.
Can I ask a quick follow-up?
Yes, of course.
In that realm of the really, really dense.
I think you mute it.
Oh, sorry. For cells that might be on top of each other or slightly off, it can be really hard to delineate edges, particularly with cell segmentation. Like how, I guess, what do you do in that scenario?
I see. Yeah, that's an excellent question. And in your case, when the cells are physically on top of each other or even overlapping, in that particular case, because we're representing cells as points in space rather than the segmentations, we would consider these cells neighbors, but yeah, we would just consider these cells neighbors and somewhat ignore the fact that they're kind of on top of each other. whereas, two cells can be really far apart, and, we can see their peripheries that are not actually touching each other, but we would also still consider them adjacent. We'd still consider them spatial neighbors with each other. But you're right, in this particular case, we don't require spatial neighbors to mean actually physically touching each other.
Cool, thank you. Great,
great questions. Let's see, and let me just go down the list then. So for Kun Kyo Yang asks, can meringue be used in subcellular level organization? That's an interesting question. We haven't looked into that, and I would, I think for subcellular level organization, it's because we're looking at spatial cross-correlation patterns or spatial autocorrelation patterns. There needs to be variation in the thing that you're correlating. So genes have a large dynamic range in terms of their values. So you can see it's high in some regions, low in some regions. Subcellular, I guess when I think of subcellular, I think of molecules as either being here or there. So that to me sounds more like these type of neighbor enrichment analysis. But I guess if you have subcellular level information that's like in this region, this gene expression is higher, or there's some way to get more dynamic range in terms of that expression level. I suppose it could be used. But yeah, I think I would have to think a bit more about the particular question or the particular context, and whether the spatial autocorrelation would give anything meaningful there. See, then the next question, Salai Taman says, nice talk. Is meringue able to infer cell-cell communication of non-adjacent neighboring cells? In theory, as I mentioned, this adjacency weight matrix is quite flexible. So if there's some way for you to systematically annotate interacting cells that are not spatially adjacent, in theory, we could look for spatial cross-correlation patterns among non-adjacent neighboring cells. But yeah, I think it's just a matter. I think it's a matter of can you define which cells you want to test as communicating in some systematic, unbiased way, I suppose. But yeah, I think it's fully amenable as long as it's just about defining that spatial adjacency relationship. Let's see. And could you perform the meringue spatial organization on RNA spots directly and avoid segmenting into cells completely? This is a question from Caleb Stoltzfus. And it's a really interesting question. Again, I think because we're relying on variation in gene expression, it can't just, I guess it's going to be a little hard if it's just on or off to compute correlations that are driven by all zeros or all ones and ones. So I think it logically makes more sense to me if things are segmented into cells. But I don't know. I guess I would have to think a bit more about what type of questions I guess could be addressed by looking for spatial cross-correlations among RNA spots, because there are also other approaches such as you might want to ask for this RNA species, is there enrichment of different if you look among its spatial neighbors, is there an enrichment of a particular RNA species? That question can be addressed using other hypergeometric tests or spatial neighbor enrichment analyses. So I don't suggest that meringue is going to be the tool for all types of analyses. We still have to pick the appropriate analysis method for our question of interest. So I think generally, for depending on the question that you're interested in with RNA spots, another type of analysis might be more appropriate. Let's see. And then I guess Edward has his hand up.
Hey, so I had just a couple more questions about using meringue for ST or Visium data. So what are the benefits of meringue for ST or Visium data over something that directly models the gene expression? Like, does a regression model for the gene expression like spatial DE or
yeah, that's a great question. We actually do compare our approach to both of those in the paper. And for Visium data, because you are dealing with uniform density pixels,
exactly,
the results are very, very highly correlated. I would say there's very little difference. The only potential difference is just speed. We coded meringue in C++, and it's a lot more memory and a lot more time efficient in terms of scaling with respect to the number of cells and a number of genes. So you might have some minor advantages there in terms of speed and memory usage, but overall, all the three methods should give you roughly the same results.
Okay, yeah, that's good then. And my second question is, a lot of times when we are looking for spatial expression patterns, we're more interested in seeing whether the variation is within a cell type, right? Because that, if one side is tumors and the other side is T cells, they're going to have different expression. That's not super interesting. So I guess, can we use meringue, or do you have any other ideas for how we can identify spatial expression patterns within a cell type?
Yeah, that's a great question. And that's, I think, something we tried to tackle with the MPOA analysis, where we complemented meringue with more traditional clustering analysis, where we first do clustering to identify the cell types, and then within a particular cell type, we can I try to look for additional spatial patterns. So that's the way I would go about doing it. To first do clustering analysis to identify your cell types, and then for the pixels that correspond to T cells, see if you restrict analysis to just those pixels, can you see spatial gradients or any type of spatial variation?
So we should show that a little bit in the etc. neurons, right? So in your talk, you can
yeah, exactly. When we limit it to just the E8 neurons or the I11 and I6, just within those subtypes of neurons, you can see spatial gradients.
Yeah, I think Liang Ding also asked more or less the same question, so 10x Visium. But there's another question about the mRNAs. That's more computational algorithm. So if you can rate that.
Yeah, sure. Let's see. Joey Arthur asks, why does Moran's I inflate type 1 error with distance-based W matrices? Is there an assumption going into variance calculation that's being broken? So let's see. So why does Moran's I okay, it should be, it inflates the distance, it inflates the type 1 error rate if we have this particular situation where we have variable cell density. So it ends up misinterpreting so you can have, for example, uniform gene expression, but because cells are denser in some regions than others, it ends up getting picked up as spatial autocorrelation. So it has to do with, it has to do with let's see, is there an assumption going into the variance calculation that's being broken down? yeah, I don't know for that. I would have to, yeah, I would have to double check on the variance calculation specifically. But yeah, I think it is it is a particular situation for the Marazi calculation that might be a little bit unique to cells within tissues. Okay, and then let's see. Yun Chen Gang asks. Communication between cell types is done in a pairwise manner. Did you try to analyze multiple type communication, for example, simultaneous interactions between B cells, T cells, and macrophages? That's a great question. No, we did not. So you can see in the spatial autocorrelation statistic, it's or the spatial intercell type spatial cross correlation statistic. And we only have X and Y. I guess in theory, we could have another variable in there to have, expression in the third cell type. But yeah, I think for that, I'd be, I'd wonder, are there certain interactions that are, I guess epistasis is not the right word, but like somehow only present, let's say, between T cells and B cells when macrophages are present or something like that. And that becomes a lot more complicated than just, cross correlation. So it's going to be essentially, a more of a posterior correlation given a prior of the presence of a macrophage or something like that. So I think once you go beyond pairs, it can get a lot more complicated in terms of the interaction terms. So I think it's a really interesting question. I think, yeah, it's definitely something we should continue thinking about.
Great. So I was going to also highlight the fact that you're using geospatial algorithms are great, right? Because I recently see astronomy and imaging, right? There's a new recent paper studying tumors. And now I think the interdisciplinary work between different fields, right? Geology, astronomy, sociology will actually help spatial biology in general. I think
absolutely.
I'd like to highlight that from your great work. And also, we should also right now, Jeff works hard, I guess, because you already presented very impressive data. Thank you so much. Are there any other questions?
I think I see a hand from Coles Price.
Hi, yeah, I had a kind of quick question on the glioblastoma work. So as I remembered from your presentation and how you guys defined and looked at cell states, originally kind of defined from previous single cell work. Now that we're with some of these analysis tools, now that we're getting at this is kind of a weird question, just to full disclosure. Now that we're getting better and better at detecting cell states, even more resolved than the ones we kind of defined before, how well do you see these tools on, especially particularly, say, the cancer fields where cell states are dysregulated, they're dynamic and kind of messed up because of the cancer state? How well do you think we're getting towards predicting or maybe predicting transitions between cell states? Like instead of because we're always taking a snapshot of when we section the tumors, are we getting closer enough to saying like, okay, we define this cell state, but we have indications that it's transitioning to this or on its way to potentially becoming this?
Yeah, so I guess the question I have for you is, are you familiar with RNA velocity analysis?
Sorry, go ahead.
But yeah, I think that could be an interesting way to look for, as you've said, these are static snapshots. So can we try to predict that, the cells of this subtype are actually upregulating genes that are making them look more like cells of this subtype, and perhaps, in the future they will actually transition. I think, there's also if you're interested, I think velocity analysis might help with that type of prediction. But I think I'd also just want to emphasize that there's going to be some a limitation to the extent to which we can, computationally predict things. And at some level, we're going to have to, go back and just validate and perturb and observe the subtype transition and see if that actually occurs, within a model organism, for example.
Yeah. No, that's a good. It's a good point. I guess I was just wondering about. Been reading a little bit more about temporal snapshots, essentially, and whether it's almost like looking inside a cell with subcellular spatial resolution. You kind of can get snapshots of time of what maybe that cell was like before, and
definitely a really interesting question. And maybe one day Rong will create temporally resolved spatial transcriptomics and help us answer that question.
Oh, thank you.
I was about to ask you that question, actually, Jean, but I think you have the right tool, which is RNA velocity. But on top of that, I think probably if you can look at multiple layers of the omics and see how epigenetic regulators kind of prime the cells to become more kind of mature or more differentiated, then that will help you understand better as well. So that's kind of, I had a question, but so many questions in the chat box, and the final I think I can quickly ask. So in your accessory neuron data analysis, you see kind of within that just one type of cell, you see some spatial heterogeneous distribution, right? So have you done RNA velocity analysis? Is it indeed actually that's the differentiation process? So just some of those accessory neurons are more kind of early or progenitor accessory neurons. Some of them are just more mature. You see they're actually spatial distinct, or spatial differential distributed.
Yeah, that's a really excellent question, and something that we're frankly still struggling with to address. Essentially, that so essentially, what we would love to do is RNA velocity analysis within tissues and try to see as exactly as you suggested, if certain cell types are differentiating towards others, or there's some type of relationships we can define between the subtypes of neurons in that manner. I think it's definitely something we're working on. Turns out segmentation in tissues or velocity analysis within neurons is quite challenging, given there's just a lot of segmentation challenges. But definitely a great question. Definitely something I think worth looking into.
Exciting. Any more questions, Rong, or should we? Okay, so if there's no further questions, I think let's thanks to the speaker one more time. I'm going to use this clapping, and great work, Jean. I think this was fantastic. And on your black backgrounds and cute images, they're really nice in terms of presentation style.
Oh, great.
And before we also wrap up, I'd like to highlight that next week we will have Dr. Akil Merchant from Cedars-Sinai, Los Angeles, presenting his work on imaging mass cytometry. And the title will be Single Cell Architecture of the Immune Microenvironment in Lymphoma. And with this, then we'd like to wrap up for today. And please enjoy your weekend, and hope to see you guys next week.
Okay. Thank you, everyone.
Thank you. Bye-bye.
Thank you. Have a good weekend.
Take care.